So our next speaker is Dr. Dick Shaw from CABI. He's the regional director of CABI here in the UK, I think. And he brings a wealth of information on basically his whole career is dedicated to biological control. So I think we can look forward to hearing from him what the latest developments are in this area. Thank you.
Thank you, and thanks to the organisers for inviting me and feeding me and housing me. It's a joy to be here. I didn't come the last time. My colleague Kate Pollard came 3 years ago and spoke about Himalayan balsam, which I can update as well. But my task today is to update you on the biocontrol projects that we have currently ongoing that are in various phases of post-release to early research, and update you on how they're getting on, and a little bit more information as we go along. So we've got an excellent team of staff at CABI in Egham, based in our Egham Centre, where we have 2 quarantine facilities, propagation, everything else. We normally have a couple of interns each year. Watch our website for adverts if you've got any colleagues that might want to come along for a summer. And we've just successfully recruited a new member of staff who starts in January. So a growing team, the growing workload, because the plants are growing and we're not stopping them coming. So we have a range of skills across entomology and pathology. But CABI is a not-for-profit organisation, mostly international development, helping farmers lose less in developing countries. So this is a bit of an outlier to what CABI does, but we're good at it, and we're probably the biggest team in the world doing it outside of the US. So we don't just work on UK weeds. The batch of UK weeds are up at the top there, you can see. But we have an awful lot of other weeds from much more exciting places around the world. So along with our Swiss team, who have another 27 targets, we're doing an awful lot of weed biocontrol around the world. So this is not new to us. We've been doing this since the beginning of CABI in 1910. But the stuff in the UK and Europe, the UK is way ahead of the rest of Europe, don't forget, as far as invasive weed biocontrol is concerned, is relatively novel here. We've got a paper at the moment just in review looking at public perceptions of biological control of weeds, basically showing a difference between Europe and America, or Europe and the rest of the world, in understanding, experience, ability to cite examples, and gut feeling. And in general, Europeans don't like the idea, and everywhere else they're kind of well used to it. And I think that's to do with getting a success under your belt, proving it works, and people becoming trusting of the technology. So we're also doing microherbicides. I'll come to that in a moment. So yes, at the moment we've done 4 classical biocontrol agents released in the field. So these are the first 3 and then the 4th in Europe, the first ever. And we're also looking at new targets. So we've been consulting our stakeholders, thanks to funding from DEFRA and Natural England, and also looking at the costs, updating our cost paper that we did on invasive species to GB, and that's very shortly to be published, I think. So yeah, we're quite active around there. And the principle of what I'm talking about today is classical biological control, mainly. I'll talk about microherbicides as well. But the idea, the principle, for those who haven't listened to me bore you before, is you've got an invasive plant population that's too high, be it economically or ecologically, whatever you choose. There's a threshold you want to get it below. You introduce your biocontrol agent, and that line can be incredibly wide or very short, depending on how lucky you are. And then you get a boom in population and a suppression of the target, and therefore a suppression of the natural enemy that is only dependent on the host to be alive, basically. So you end up with a suppression, which is an equilibrium that goes off into the coffee room. So that's the principle of what we're operating on, to get it below that threshold. And I've been doing it for a while on Japanese knotweed, which all of you are very familiar of. And since you're all still in work, it obviously isn't working quite as well as I was hoping. But I'll explain a little bit about why that is. So here we have our psyllids, Aphilara itadori, named after Japanese knotweed, itadori. Our colleague Daisuke has been working on this, and he's just got married last week, and he's not back yet, so he would have been here as well. But that's the psyllid, sapsucker. And over 12 years of a research program, we started talking about this in '89. Then we started working on it in 2000, and then we got a release in 2010. A 5-year monitoring program all over the country. It's the first biocontrol agent in the world that had a contingency plan attached to it. So that's how concerned we were as a nation about the idea of what might go wrong. So we went along with that, released it, and kept releasing it and releasing it and releasing it and releasing it. And then various things happened, but it never really established. So the bottom line, which bullet I didn't want to put in, is it's not working, which is basically what's happening. But we are still working on it, and our colleagues in Canada And the US are still releasing and monitoring because they're still quite hopeful, and we're also speaking to the Netherlands. But what we started looking at was why is this not working? So the principle we work on in biocontrol is safety first, always. So what we did was we went back to the native range in Japan and work out exactly where that original strain came from, where Dr. von Siebold, the cause of all our problems, was based in near Mount Aso, in Fukuoka, near Fukuoka. So that's where he got it, and that's the clone we got. So we went straight there, and that's where we ended up with John Bailey's help to work out where to go. So we wanted a match to our clonal knotweed. We didn't want something that was from somewhere else that wasn't likely to attack it or attack other things. So we focused rather too narrowly, with hindsight, on matching the host with the natural enemy. When we did some climatic matching, Corinne Pratt did this work. You can see this is where the range we should be looking at for the UK or for GB. And what we were doing is climbing up a mountain in semi-tropical an area, this is very hot down here, to find a similar match to the UK, which is not how you do it really, with hindsight. So we started to look back up here again, although we did survey there many years ago. And, well, I'm just summarising again. We found a psyllid in Murakami up here on the left with some interesting damage, like curling damage, I'll show you in a second. So this was much better climatically matched. We brought it back, and we had to make sure it was the same psyllid, so we did lots of tests on it to prove that it's exactly the same species. We had to show that it wasn't carrying any parasites or the usual things that can be vectored by psyllids. And finally, we could convince the government that was a good idea, and we got permission to release in 2021, very early on in 21. So this is really new stuff. And we only released in 2 sites. We didn't want to swamp the population with what we've already released. We wanted to see how it was going to go. So we had a release on Bohemica because our studies showed us that it liked Bohemica much more than Japonica, unfortunately. So we did that study, we showed that, and sure enough, that's what happened in the field. So we've released on a Bohemica site and a Japonica site. And whilst we have damage at both, the Bohemica site is much more distinctive damage. So that was quite encouraging for us. The Dutch got wind of it. We got some funding from them to support the work, and they've been releasing in the Netherlands, and they've got way more Bohemica than we have. So it's quite possible, unfortunately, that your taxpayer dollar has gone to support possible biocontrol success in the Netherlands and mainland Europe rather than the UK. But let's see. Let's hope one of them works. So this is what we're seeing in the field now. This is not lab stuff. This is field work. So that's the sort of damage you get. This weird curling. And when you unpeel the curl, there's your little nymph sitting in there sucking the sap. So it's a physiological reaction to the organism feeding on it. You get this on peach and other things with some aphids. So you can see this sort of thing around the world with different things. It's not a vector disease. That's the important thing. So in the Netherlands, so we're seeing this in the field. So I'm super excited about this, obviously, but it's Bohemica. But in the Netherlands, you can see this is in the field again. This is all over the field site that they released at. And that's what happens when you put a sleeve on it. So it's absolutely mullered, and I'd love to see this in the field, believe me, but obviously that's with a sleeve on it, so it's got an artificial habitat, no predators, all the other stuff, so it's got a big advantage. So effectively, watch this space. This is really early stuff. We had a Canadian visitor come over, work with us for a year to work out how is this curling happening, and he's publishing that paper very shortly, so exciting development. Meanwhile, I shouldn't forget, we're still working on a mycoherbicide, so part of our project work, check my watch and make sure I'm not going too fast. Good. Part of our project work was to find natural enemies. So we found 186 species of insect and 44 pathogens feeding on Japanese knotweed in Japan, with endophytes and all the other things that are living with it. But you find nothing on it here, obviously, very little. Part of it was this Mycosphaerella. So if you look, you could spot knotweed in amongst Houtinia. There we are, there's another one sitting next to it. You can only see the knotweed because it's got red blobs on it because of this Mycosphaerella pathogen. So it's very, very distinctive, very common, ubiquitous across Japan. Unfortunately, it's got 2 hosts and the life cycle. Unfortunately, no, that's not true. It has the same host that Japanese knotweed, but it also can infect a very rare native species of Polygonum. So that was it. Sink it. We couldn't possibly release that. What we discovered, partly by accident, was that you can infect plants with mycelia. So normally it's the spore that flies off and lands on your plant and then hits your non-target. That's no good. But mycelia, you could almost brush the mycelia onto the plant and it will infect and create the same cycle. And we also discovered that it has mating types, male and female, plus or minus it's called. If you only have one mating type and you put those mycelia on the plant, you cannot ever get to a spore. It's impossible unless someone brings in the other mating type. So, okay, maybe we've got something that is specific to Japanese knotweed, can be put in a bottle and can be sprayed onto the plant and infect and cause this damage. So that's what we thought, wow, this is exciting. I won't tell the whole story, but anyway, we discovered that we were 7 days away from having revealed the information before we could get a patent. So we had 7 days to write a full patent for the European patent application, which was a terrifying week. And we successfully secured a patent for Europe and various countries, and we're going to the US. We've given up, but Canada, we're going to get a patent there as well. So this is great potential, but it's not a thing in a bottle yet. There's a long way to go. So these things take a lot of work, and it's also beyond our skill set. Like, we're pathologists or entomologists. We can get to the point of this has got potential and show field trials, but getting it to work is a very different step. So that's what we're looking at industry partnership at the moment. So who knows, you might have a not weed specific bioherbicide available in the future, which I imagine would have quite a big market. And my MBE will be waiting in the post. So anyway, not that I did discover it. So onto Himalayan balsam. We all know this one, beautiful but deadly. Native to Himalayan foothills, tallest, biggest nectar producer, amazing species, famously invasive. There's not many places that don't have it. And we went back to the Himalayas and we found this rust fungus, which we had to name. It was a new species, so quite often with biocontrol you discover species that are new to science. And what it does is a double whammy. It hits the seedlings first with these etial cups, and the seedling etiolates way above the rest of its host, its neighbours and family. It sporulates and lands upon them, and then completes its life cycle through the spores, which is the mass production sector of the pathogen, and goes on to infect more and more. So what have we done with that? Well, what has happened, as often happens in biocontrol, is our pathogen has detected variation in the population. It's a sexually reproducing plant, so it's potentially not surprising, suggesting we've had multiple introductions. It's not just one clone or one population that's evolved, it's lots of different introductions. We think at least 3 from the R&R sites in Himalayas and India, where basically people collected plants and brought them back during their tour. So we brought this back, and we did lots and lots of studies on the molecular makeup of the knotweed around the country. And there's one big clone, which you had presented 3 years ago, which is kind of clade 3, and then there's 1 and 2. So we've got the pathogen that hits 1 and 2, which is Pakistan and Indian strains of the pathogen. And we desperately needed, as Carol Ellison, who passed away a few years ago, said, we need to get to Kashmir. And at the time, you did not go to Kashmir, no one went to Kashmir. So we were extremely frustrated until 21, when we finally got a shipment back from Kashmir, which has successfully infected that clone in our collection. So we're really excited about what this could mean. It means that we can have the same effects across the country rather than just in the susceptible sites. At the moment, we have to say, someone says we've got a problem and we've got a small budget to do it. We have to take the seed, bring it back, grow the plant, find out if it's susceptible, and then release, which is really time consuming, but we don't want to waste anyone's time by showing that, release it and nothing happens, because then you get a bad reputation. Unfortunately, the Canadian populations are not susceptible to any of them, so thankfully, well, most of them, so thankfully we don't have their strains. So yeah, we've got stuff happening, it's spreading locally, and we're doing a lot of work with local action groups who are helping us distribute this and get it in the field, so we can't be all over the country wasting it. Well, it'd be a waste of our time and your money to do that. And we've also developed a really nice app, open data source app, which is really handy for us to help monitor and help the people. And we've been evolving this because sometimes you ask pointless questions that people don't answer and you waste their time too. So that's being refined very nicely. And the other issue we have is what you tend to do is tell people they've got a problem, then they say, oh yes, we'd love to be a site for a release, and then they go and pull it, or someone gets excited about it and pulls it. So we've lost quite a few sites as a result of over-enthusiastic management, which is fair enough. Onto Crushula. So Australian swamp stonecrop, we know it's from Australia, not New Zealand, or it used to be called New Zealand pygmy weed. We did the molecular work and it's definitely Australia. Introduced widely, seems to be able to recognise what sort of status your site has. If it's Ramsar and triple SI, it will get in there. And it's very, very invasive indeed. Challenge for us is it's submerged and emergent. And there's a big debate about, in my opinion, a debate about what its normal habitat is. In Australia, I think it's a terrestrial plant that gets inundated on occasion, whereas here it's a kind of aquatic plant that emerges, in my opinion. So it's an interesting way the plant has behaved in a different country. So we went back to Australia and found this mite, Aculus crushulae, beautiful thing, and it is unbelievably small. I can't explain how small this thing is. So this is a crushula tip. So you know how small crushula is, and these are hundreds and hundreds of nymphs on there. And how so small is it that I was asked by Sonal to collect some clean material. So as an entomologist, I went to Australia and we collected various bits. And I had one particular mission where I went to the cleanest patch, collected these tiny samples in little vials, cleanest possible. That was the origin of our culture, because it was in there and I couldn't see it. So incredible little thing. Lives on the terrestrial stuff. It can get inundated. I think it survived 4 months where the site was underwater at one site, which I find extraordinary for something that isn't supposed to be able to breathe underwater. But it's not going to survive long-term inundation, which has been our main challenge this last few years. And this is the sort of damage it causes over here. So this is what the plant normally grows like. And then you get these stumpy little buds that are like galls, basically. And if that was a tree, you'd be freaking out, wouldn't you? But because it's a tiny little plant, no one thinks that's impressive. But I think that's quite an impressive reduction. And there's a more realistic one, both set up at the same time, with the mite, without the mite. So it certainly has impact. And we've got sites, I think we've done 20 sites so far, up to 12 in 22. Because it's a controlled plant, we have to collect the plant from the site we want to release at, bring it back to our labs, infect it, grow it up, and then replant that individual thing to the right place. So we have to keep all of these things separate. It's a bit ridiculous, really, but I can understand why that is. So they have to be on live material, and then they start to spread. So it's been slow, the spread. We've had a lot of problematic weather, as we all know. But it's certainly establishing in the field, and you can see damage in the field. So this is early stage stuff. And then floating pennywort, probably our most exciting project. Amazingly fast-growing plant, as you know, 20 centimetres a day recorded. It's just, you can watch it grow. And these impenetrable mats, so you could come back in 2 days and that's completely covered and non-navigable. So this is a really big threat to the country and the rest of Europe too. Sorry, it's native to South America, in particular Argentina, across the range of Argentina. Our weevil, Listerionotus elongatus, was identified in the 70s by a guy called Hugo Cordo, who spotted it and said, I bet this plant's going to be invasive, and I bet this agent's a good one. That's how talented this man was. And sure enough, the plant became invasive, and sure enough, this is a cracking agent. So there's the adult weevil. It lays eggs, shoves them into the stem underneath. PTL, larva emerges, and then mines. It doesn't emerge from the plant. It needs to stay in the plant. Mines the stolon, pupates, and then out comes the adult for next year. You should be able to get 2 or more generations in the UK, which is really good because that helps you really hit the plant. They're bulletproof, these things. You can step on them and they're pretty tough. No, you can't step on them. You can try and squish them. And they're also beautiful. So we've got some new camera kit and we're quite excited about it. So a lovely image of a weevil chowing down. So we've released it, I think it's 7 sites, maybe more actually. Jamie Jador's running this project, and we're working with the paddlers and other people in the field to get the things out there and monitor them. We've showed that it can overwinter successfully in the glasshouse and the field, and it's starting to spread. This is the first year of proper releases, and I can't show you the picture because I haven't presented it to the government and the donors yet, but it's really exciting. So very impressive, understated damage. I can't tell you how good this looks in the field. So we don't want to shout about it too much because it might all crash out in the winter this year, who knows. But we're hopeful that this is enough to get the population going properly. And if you get it synchronized correctly, they emerge in the spring and out we go again. Sometimes you can be out of sync and that can be a problem. So watch this space. This is one of our big stories, I think. 13 field releases, 2,300 weevils. They're all got to be hand-picked. It's really quite hard work. And we're overwintering in this lovely polytunnel. And then finally, I shouldn't forget Azolla. So the Azolla weevils are still out there. We're still using it. And we did a really nice paper recently. Corinne Pratt, this has been around since 1911 or whatever, 21, and it's been used and we spread it around and it works amazingly well. This is a triple SI in Surrey. And that's what it looks like after control. So it really is, you normally see the other way around, don't you? It looks like that and then it gets invaded, but this is the way around it is. So really good weevil. It eradicates its host. Biocontrol agents aren't supposed to do that, but I've talked about that before. But we've done a bit of economics on it. So the weevil is saving the country between 8 and 16 million a year. And the augmentation program knocks it back down again. So just the augmentation program alone, which we do, has got a benefit ratio of between 44 and 88 to 1. So really good investment. Any economist will tell you you rip your arm off for that sort of return. So quite exciting. We're not stopping there. So the UK again has quite a good vision. They're looking at what the horizon scanning things, what things are getting out of hand that need control, and is there a biocontrol solution? So can we have a quick look and see if it's going to be suitable for that? Myricum aquaticum is obviously one that we're all very aware of, very problematic plant, emergent aquatic. And we're no strangers to biocontrol. There's been a couple of agents looked at quite closely by colleagues, so we're not picking up from the beginning. This is piggybacking someone else's work. We've got the Lysathia beetle here. That's in our culture in quarantine at the moment. We're studying that and rearing it. And we're also hoping to get hold of Listernotus, which I think holds more potential. It's a stem miner. So again, watch this space. And then we've got other new targets. I should have put some pictures in for you, but Elodia, we're looking at feasibility because there was a story quite regularly at one of these conferences, actually. I was brought to my attention that there's been dieback, local dieback of Elodia, and it's probably almost certainly some sort of organism, biocontrol agent that's arrived accidentally or switched onto its host. So we're looking for that. And we're also looking to play with our Morley-Toff machine to see if we can differentiate these species really quickly without having to do molecular work. And then Carpobrotis edulis, ice plant. This has been biologically trolled in the past. It's a South African species. It invaded California. They released a mealybug, scale insect or mealybug, scale insect to control it. Very successful, but it looked ugly. So people got quite upset about it and they brought in a parasitoid to control the scale, and now they're screwed. So they can't do anything now because they've controlled the thing that was controlling it. So now they're back to a square one. We haven't made that mistake, but the scale has arrived and has been found on the Scillies, and somewhere else, a Corin has been out looking for it to see whether it can be redistributed if it's the same scale. So this is quite an exciting one. It's one of those things that wouldn't necessarily come up in your massive list of targets for weed biocontrol, but because it's so impactful in really fragile habitats, it probably gets elevated up there to say, well, this is a good target and it's worth investing in. We also heard about some damage in St. Helena, so our colleagues have just come back from there, and hopefully we've brought some back. I don't know whether they have or not. Buddleia, controversial. So this came out in our list as one of those species, as we all know, butterfly bush and everything else, but it's a major problem, and the New Zealanders have controlled it very effectively with a weevil, a clear pine weevil from China. So they're not bothered. You know, they see it as a problem to their forestry and their native habitat, and they'll release a biocontrol agent against it. Whereas for us, it's like, oh, it's a weird butterfly bush. The gardeners love it. Doesn't really have that much value outside of nectar collection. So we're looking at the feasibility of it, which will probably be more about public opinion rather than feasibility, because we know the feasibility is high. The weevil controls it. So is that worth looking at it, or should we be looking at a mycoherbicide? So that's potentially where we should be going. Ailanthus altissima, tree of heaven, is another one. There's a really nice eriophyid mite, very similar to the mite I showed you with Crassula, that's caught up with it in the Mediterranean and is causing quite significant damage. So there might be other mites, or that mite could be potentially, if it can be climatically matched to us. So there may be something on the horizon for Ailanthus. Not a big issue now, but if you see coming into London, it just transfers from Buddleia to Ailanthus as you get close to the city. So it will be coming, and it will be getting much worse. If you go to Italy or France, it basically does what Buddleia does all over the railway. So similar plant. And then finally, the old rhododendron, mainly looking at Chondrosterum purpureum. I'm still interested in whether there are seed feeders, because the old literature, when it was first introduced, talked about a multitude of of beasties or weevils or whatever they described it as coming out of the seed collection. So there could be a seed feeder which would be publicly acceptable. You let it flower, everyone goes, ooh, and then you let the seed get taken out and it doesn't spread so much. So we'll see. I think we'll probably look more at this stump treatment, the idea of a silverleaf pathogen. And then finally, Ludwigia. This is not a big issue for the UK because we've got an eradication programme on the way, but this is massive for Europe. And it's got these lovely agents sitting there because the Americans have been looking at it as an invasive, much the same as the Nymphaea species you're looking at. They've got a biocontrol program against that because they're ahead of us. They've got a bigger problem and had it for longer. So we're still hopeful, but they're really resistant in France. Really, it's just amazingly difficult to get them to listen to anything other than doing what they're already doing. So hopefully we'll get there, but we've been at this for 10 years and no one's listening yet. So yeah, that's a quick summary, I think. And there's a website where we can go and see some more updates. And we produce a public update for the biocontrol projects every 6 months. So look out for that. And if you want it, just go to our website. It's also on the Non-Native Species Secretariat website that Olaf looks after. And I think that should be it. Thanks to everyone. I tried to put a slide together with all the people we partner, and I couldn't get it on 2 slides. So you're all in it, and everyone else is in it. So thank you.